Good afternoon, everyone, and welcome to the CCOVI Lecture Series. My name is Jennifer Kelly, and I'm a postdoctoral fellow here at CCOVI, and I'm here to introduce our speaker today, Dr. Jeff Stewart. Jeff attained his PhD in zoology at the University of Guelph in 1998. He went on to do a postdoctoral fellowship in biochemistry at the University of Cambridge. In addition to that, he did further postdoctoral work at the Institute of Aging at NIH in the States. Dr. Stewart joined Brock in 2004 and has been a CCOVI Fellow since 2010. His research is funded by NSERC, the Ontario Centres of Excellence, and the National Institutes of Health. Dr. Stewart's research focuses on mitochondrial form and function, including how these are affected by natural products. Today, Dr. Stewart will speak to us about a green extraction method for polyphenols from winter grape pomace.
Thank you very much for the introduction. And thanks for coming, everybody. So I'm going to talk to you today about this one project that I'm doing with Steve Mirza and a local company called Sweet and Sticky. As you might have gathered from my introduction, this is not my career trajectory didn't take me into wine or grape research. And it's my interest in this kind of thing lies in the main focus of my lab is cell physiology, really. So how cells work in your body and what happens when they don't work. Sometimes it's inherited diseases, sometimes it's more response to stresses that aren't necessarily inherited but are but lead to disease. We study a lot of cancer, cell lines, and so on. But we are interested in natural products. So right now I'm editing this. If it's of interest to anybody, come and speak to me after the talk. But we're really interested in how different natural products actually affect mitochondrial form and function. We kind of got into that with resveratrol, which is mitochondria are a main downstream target of resveratrol, but also of other polyphenolic compounds like rutin, and there's quite a long list and growing list as people do more and more work on this kind of thing. So this is where I come from, and the main thrust of what we do in my group is mitochondrial form and function. But we started, through our interest in resveratrol, we started a partnership with Steve Mirza, who I think a lot of you might know, and his company, Sweet and Sticky. And this work was funded independently from the rest of our work, so NSERC Engage, Ontario Centres of Excellence, and also Ontario Genomics helped us with this, with what we were doing here. You'll see that it's metabolomics work that they come in with. So genomics and transcriptomics and proteomics, but also all of the omics, including metabolomics and even lipidomics, they'll fund. So Steve makes this product. I guess he is the inventor of this product called ice syrup. And ice syrup is made at the same time of year as ice wine would be made from, and it's made similarly, harvesting grapes in their frozen state. But it's this non-alcoholic product. It's more of a dessert syrup, something you would put on a variety of foods. So it's not fermented. The grapes are harvested and they're brought in and they're processed and so on. And the juices are what the product's made from. And what's left is pomace, garbage. So actually, his process alone, so this company alone would produce several tons of garbage, of pomace, every year. Every cycle of harvesting and making things. And that garbage, I mean, it's compostable. There's nothing toxic in there, and so it's fine to use as for different purposes and maybe allow it to compost, and it can be a fertilizer. But it's also got some value, probably. And the value comes in some of the things that are present in that material. And that includes probably the ones, again, you know most about, polyphenols are, that term is refers to a category of molecules, a type of molecule. And it includes still beans, which in turn include the things that you're probably most familiar with. Anybody that works in grape and wine will know about resveratrol. So that resveratrol has been measured everywhere. And we know that it's fairly plentiful in pomace, which is skin and seeds that are left over. So the grapes have been pressed, the juices removed, and if you look at that pomace, you've got all these things hanging around still. There is some value here. I was walking through a mall in Toronto area the other day, a few weeks ago, and there's a company that has resveratrol. I think it's a cosmetic company that has resveratrol in the name. It's called Resveralife or something like that. But that molecule alone, resveratrol, it's out there. People are using it for everything, cosmetics, and I mean, some of that justified and some of it questionable. But in terms of, I guess, value, people are interested and they want to pay for these things. So there's value in the sense of you've got all these different molecules that are of interest to people that are getting thrown out. Now, the other thing that we know is that some of the, these are winter harvested, late fall and winter harvested grapes. And we know that seasonally, the synthetic pathways change for producing some of these polyphenols. Again, it's been well studied, particularly in resveratrol. And resveratrol is made by, it is a still bean, so it's the still bean synthase pathway, a series of enzyme catalyzed steps that give you resveratrol and viniferins, pterostilbene and polydatin and so on. They're all made as a resveratrol backbone that gets modified by the plants into these different molecules. There's 20 to 30 of them in that family. And this is a transcriptomic, the results from a transcriptomic study just showing, so this would be a gene ontology analysis result, a GO analysis, and what they're asking there is what types of genes, and which types of genes do you see enriched expression under some set of conditions? So they've harvested grapes at different times, and they're following the transcriptional profile of the genome. So these are all the genes that the grapes are using actively, making transcripts from. And one of the highest is over here. You can see, so they're divided into categories. Regulation of the cell cycle would be a category, but this is a category. Still being very difficult to read, sorry, but that's still bean synthesis. So one of the things that happens with grapes in winter for whatever reason is that there is this upregulation, this increase in the expression of genes that are for making molecules like resveratrol. So you've got waste pomace in fairly large quantity, and we know that the synthetic pathways are probably turned on. So you'll see this is November to January, and these are increases in still bean synthesis activities at the transcriptional level. So all the enzymes are being made. So there's probably lots of resveratrol in that pomace. And resveratrol is certainly of interest, but it's the one that's been out there for the longest of the polyphenols. And if you look at the polyphenols as a more general sort of category of molecule, and polyphenols are anything that with like multiple phenolic groups, these big phenolic rings that give them their chemical properties. And there's all kinds of them. There's hundreds of polyphenols. And even though resveratrol has had the most attention, it's quite possible that many of these other polyphenols are increased seasonally in the same kind of pomace. And they're of interest. Maybe I'll flip ahead and just show you this. I just downloaded this from PubMed just for this talk. And what's shown here is this is the number of publications per year for a search on polyphenols. So if I PubMed is you may not be as familiar with PubMed, it's for health science, really, publications for medical science. And so these are polyphenols used to be a bit player, a few hundred papers published in a given year where the term polyphenol came up, and in relation to some kind of health-related phenomenon. Nowadays, if you do the same search, you'll get almost 3,000 papers a year that are coming up based where polyphenol is a key word. So people are doing these experiments, and what probably happened in large part is resveratrol was a progenitor molecule where people realized it was involved in certain, it had maybe some, it's still I think not resolved, but had some potential health benefits. And that kind of led to realizing that resveratrol is a stilbene, and there's these other resveratrol-like molecules, and it's a polyphenol. There's just a lot of effort now in that area. And if you look at the types of polyphenols that are found in wines, and particularly in red wines, you'll see that if you were to look through PubMed, there's a story attached to each of these. And the stories can be broken down to, they're more extensive than this for sure, but there's a lot of evidence for polyphenolics having some kind of role in cancer, in type 2 diabetes mellitus, and in some age-related phenomena. Maybe not aging itself, the biology of underlying aging, but age-related phenomena like neurodegeneration, metabolic disorder leading to diabetes, this type of thing. So these are all molecules that you would find in red wines. And they're not just in red wines, but they're red wine grapes. So they're not just in red wine grapes, but they're in the primarily seeds and skin. They're mainly in the pomace. And they'll make their way into the wines because they tend to be very poorly soluble in water, more soluble in ethanol, and so they will be extracted into wines during the fermentation process. So there's all this activity in terms of biomedical science in polyphenols, interest in polyphenols and research activity. And I would say it's bigger than just scientists. These are making their way into the broader public. And one of the things that has come up just recently while we were doing this is more of an emphasis on quercetin. So quercetin is the new resveratrol maybe, and there's increasingly interest in that molecule. In my field, where we are interested in cell behavior, the biology of individual cells, and we're interested in aging. This idea has come about of senescence, cellular senescence, and more recently, senolytics. And that has actually made its way just recently into the popular press, so even Time magazine will talk about this on occasion. It's based on the idea that in your body you have all kinds of all of your tissues, all of your organs have resident stem cells, even as an adult. You think about stem cells as being something that are present in really young people and during development. But even as a fully physically mature adult, all of your heart, your brain, etc., these stem cells are present. You've got tissue resident stem cells, and they're there for the purpose of regenerating tissues. And those senesce with age. And what happens is when they senesce, they adopt this different phenotype, and they begin to secrete a variety of pro-inflammatory molecules. And that's called the senescence-associated secretory phenotype. So senescence is the cessation of dividing. So you're inherently at that point not going to do your job in replacing cells, and that's aging. But on top of that, they begin to pollute the neighborhood, really. So they're making these molecules that attract white blood cells and inflammation, low-grade inflammation, to different parts of your body. So someone had the idea that, if you could go in and remove those cells, then, you could alleviate some of the phenotype, some of what's going on in human aging. And so that became a field of study. And one of the first molecules that was, so the way people do this kind of thing is they do high throughput screens. So they've got cells growing in dishes and they fire a million different things at them. And they're looking for something that reverses this phenotype, this senescence phenotype. And one of the first things that came up was this product that's in this polyphenol that's in red wine called quercetin. And we've known about quercetin for a long time. It's been studied in other contexts as well. And really, I would say what's happened is identifying it as a senolytic might be just attaching a mechanism to previous observations of its benefit in terms of anti-cancer effects and so on. So it's a senolytic, and what that means is what they showed first in cell culture, but they extended that into live animals, was that you could deliver that quercetin, and actually often in combination with other polyphenolics that are not necessarily plentiful in red wine. And whereas nothing would happen to normal cells, it would cause these senescent cells to die. And so the lytic part is they're dying by apoptosis, so they're breaking up, a controlled cell death. And that's kind of what you want. So it surprised me when that first came out, but it has stood the test of time, short amount of time, 5 years, and then extendable into animal models. So you can see, so now all of the age-related phenomena, whether it's vision or function of a heart or cardiovascular system generally, these have all been studied now in mice, rats, plus or minus quercetin, and you can see these effects. And if you go in and look, you'll see that there is evidence that these cells are cleared, these senescent cells are cleared from the body. So that's become a big thing, a big field of study. So all of those things, I could go on about delphinidin and camphorol and quite a number of these different polyphenolics. They all have stories like that attached to them. But that's really where the value lies, I think, in the pumice. So this stuff is being thrown away. So Steve Mertz's company, Sweet and Sticky, they produce several tons maybe of that pumice a year, but a group at Carleton University did, Dwyer et al. 2014, they did this study where they tried to place a value on how much pumice is being thrown away in different parts of Canada. This is Ontario, they looked at BC as well. And according to their estimates, and they took it out as far as 2011, $500 million a year of value, pumice value is being discarded and not realized. Now, that value, in the paper, they're quite frank about the fact that that is inflated by the idea that they're saying that if you were to get all of the valuable things and people bought all of those things from pumice, that's the amount of money. It's unlikely to be that high, is it? But also that's 10 years ago almost now, and so there's definitely going to be around here more of that produced. But the bottom line is there is value. It's been estimated by non-scientists, by economists, and the value is primarily in these weird molecules, these polyphenols and some other things with a lot of them are thought to be good for you. So those are all things that I usually do and often think about. Certainly the senolytics and resveratrol and with quercetin and so on. But so with Steve Mertz's company, we went in a slightly different direction and just thought, well, as long as we know about these things, we can help that company extract the important bits from their garbage. And there's lots of ways to extract molecules like polyphenols. You saw those big phenolic rings, and those tend not to be very water soluble. So we're talking about molecules with really low water solubility generally. But also we wanted to, or Sweet and Sticky wanted to be able to extract these in an edible form. It doesn't do much good so things that are highly water insoluble, generally you can bring organic solvents to bear to increase that solubility, but then you've got problems with organic solvents. Certainly toxicity in terms of downstream making a product that could be ingested. Also, it's a small company with limited infrastructure, so we're looking for a way to do this without massive investments in equipment. So today's talk, I think, is going to be a little bit short. I'm just going to show you results from this one project that culminated in this paper that we published in Journal of Food Measurement and Characterization. What we did was just to develop a method that actually worked to give us a broad profile of polyphenolic rescue, extraction from the waste, and then to really quantify it using a metabolomic approach. So not looking at any, I say a limited polyphenolic approach. We chose about 20 to 25 that we thought should be there and abundant, and we followed them throughout the process. So the first problem we had to solve, of course, was you want an edible product, and it's got to be cheap, and it's got to be able to extract these hydrophobic polyphenols. And so we chose beta-cyclodextrin. Beta-cyclodextrin's been around for a long time, used in quite a wide variety of foods and pharmaceuticals for different purposes. And the thing about beta-cyclodextrin, it's actually just a polymer of glucose. It's a very simple molecule, not unlike cellulose or glycogen. It's a cyclic glucose polymer. And what is useful about beta-cyclodextrin is it forms these cylindrical structures where the exterior of the structure is hydrophilic, it likes water, and the interior is hydrophobic, likes greasy molecules like polyphenols. So the idea would be that polyphenols would want to, under steady state, they would want to move into those pockets and be carried as cargo inside those cylindrical molecules. But those molecules themselves are quite soluble in water, so you're taking something that's not water soluble and you're changing that property, basically. 1.8%, 1.8 grams per 100 mil, that's a lot of beta-cyclodextrin. That's the maximum solubility at room temperature, but it's a lot of beta-cyclodextrin, so you can load it up. It's super cheap. It costs very, very little, and it has this designation generally recognized as safe or GRAS, and that's an FDA designation. It just means that you don't need to jump through any further hoops to ask permission to include it in food. It's just okay to do. As long as that's pure beta-cyclodextrin, it has no known side effects. It can be toxic at high levels, as water can be. So it's a good choice in that sense. There are other cyclodextrins with greater solubility in water that we can consider, hydroxypropyl, but they're not GRAS. They're not usable as a food product. So they're loaded into this beta-cyclodextrin, hopefully during the extraction process. That would be great, but less good if they could be unloaded. They could not be unloaded. And so what we know about them is that ring, that repeating polymer of glucose, like cellulose, like some other glucose polymers, is digested by bacteria in the large intestine. And that's also a useful property. It actually provides some protection from these molecules as they transit through the upper GI tract, where you've got pHs and enzymes that are potentially going to break them down. So there's a few advantages of that cyclodextrin. And then the other problem, of course, we had to solve is how do you treat the tissue? It's a tough cell wall. The material is actually inside of that cell wall, so you've got to get it out of the actual cells that have produced the molecules. And again, there's a lot of different ways to do this, but we're looking for simple, straightforward, and generally something that would be relatively easy to do at a small industrial scale. So we chose ultrasound, ultrasonic cavitation. You've probably used sonicators in some of your undergraduate labs, certainly in graduate labs. I know a lot of your graduate students, et cetera. But it's based on these high-frequency vibrations that produce these bubbles that grow in size, and when they implode, they produce a lot of physical force that's sufficient to shear the cell wall, shear that material, and release the contents of the insides. So it is used in food processing. Other things are used as well, but a lot of the other things would include high temperatures, which is something you would want to stay away from for a molecule like resveratrol, which is sensitive to temperature, sensitive to light. So keeping it cold and bombarding it with ultrasound is actually not bad for it, not going to change it. So, so really quick, method development. For sonication, you have really two broad choices. You can use probe sonication, you can use a bath sonicator. They're both useful. And so we tried both of them, and we used just general, at this point, rather than run all the samples through the entire metabolomic analysis, we just used some of the standard, sometimes they're presented as polyphenolic assays, sometimes they're called antioxidant assays, but generally antioxidant capacity assays that are used in this field. So the FCR and ORAC assays. So we use that approach in the beginning just to see what's the difference between one way versus the other of processing the material. And what we could see is that two things. One is we found no difference between our probe sonication, which was a small laboratory scale device, and a bath sonicator, which was also a small laboratory scale device. And we did find, though, that if you sonicated longer, you did improve things. So we worked a bit with time and temperature and these different methods of sonicating. So we had worked out some of those methods, and then it was time to use those things for the actual analysis. And for that analysis, there were 3 years worth of grapes that were collected. So these were collected by Steve Mirza and his employees over 3 years, 2015 to 17, and starting in November and going as late as January, which is really just when the company is doing their thing, when conditions allow it. And those were brought here after processing. They were brought here as pumice and stored just in a -80 freezer. So really straightforward method. It's actually just a Moulinex food processor to mince the pumice up in the beginning, just to more surface area. A quick wash to get rid of water-soluble stuff. We're trying to concentrate the molecules of interest as much as we can. Centrifugation, and then we start this ultrasound. So the metabolomics actually was done under contract with this center at the University of Alberta. And they're probably the biggest metabolomics facility in Canada. But if we were to do this now, we would do it here at Brock, because with recent grants that CCOVI has got, this is being scaled up, and it's something that will be done here by Sufan and others. But for this purpose, everything was sent to Alberta for these measurements. So we did do it in 2 wines. We chose a Vidal, or I guess that was a decision Steve Mirza made, a Vidal and a Cabernet Franc. We didn't expect to find a lot of these in the Vidal, the white wine, which naturally doesn't have as much, but we did include it just to see. And so these are the results from that, and you'll see that some of these are here. You'll see a lot of not detected. And the same analysis with red wine grapes, these Cabernet Franc grapes, the yield is much better. So to make those numbers mean something, we we did this quick analysis, which was to ask, how does what we're getting in this extract that we prepare compare to what you would get in your average bottle of red wine? This is why people think about, red wine is, if they're trying to justify why they're drinking red wine, maybe because of the resveratrol content. And so those things have been measured, resveratrol and lots of other polyphenolics. And actually, it's this website. I don't know if you're all familiar with it, but it's maintained by a consortium in the European Union funded, and it's the Phenol Explorer. Do you all know this? No? So Phenol Explorer is quite useful. You can type in basically whatever phenolic you might be interested in, and it'll tell you something about how much you can expect in a wide variety of foods. And I did, of course, resveratrol, but everything, but there's lots of information out there. Someone has painstakingly gone through the literature and come up with average values and sourced it. It's really quite useful for a comparison like this. Whoops. And so that was what was done here, is actually to go to the Phenol Explorer and get the published values for what is found in red wine. And so here I just averaged together a bunch of them. And then taking those values, I compared to the values that we were extracting from our from the grape pomace, and calculated this value. How much was the amount of this extract that we had produced? So that extract is lyophilized, it's freeze-dried, so it's now a powder. So we're giving that as a weight. What's the mass of extract that you would need to get an equivalent amount of ferulic acid as would be found in a single glass of wine? So you can see that for some of these molecules that people are interested in, quercetin, you'd need 500 milligrams of that extract. Resveratrol, of course, 326. So I think the message is that we're getting appreciable amounts, enough to be probably comparable to what you would get in wine. And then, of course, the other thing that we were able to determine is just what's the effect of time of year on these levels. And these are 3 years average, so 2015, 16, 17 average to give those values. And I think that can be useful because you can see that it's not constant. So there's a bunch of different trends which are, not surprisingly, dependent on the individual molecule of interest. So chimeric acid is fading away from November until January grapes. And at the same time, this other molecule, delphinidin, which has its own story related to cancer associated with it, that one is ramping up. We can see resveratrol here was doing something interesting, so coming down a little bit, but polydatin is a product of the resveratrol backbone, so it looks like it's maybe being converted to polydatin downstream. And then quercetin, which is probably more stable. So we've got these molecules that are of interest to us, and we've got a little bit of information about how they change over the course of the season. So I said it was going to be a short talk. I wasn't kidding. That's all I really wanted to, all I think is appropriate for you guys as an audience is this part of the work, how you do this as opposed to the cell biology of it. So what we know from this work is that we can do this relatively cheaply, simple ultrasound. It's probably not high efficiency, but it doesn't really have to be when you're talking about many, many tons of garbage, more or less. Getting some value from it is okay, as long as it can be done using a cheap process. So extraction into beta-cyclodextrin can give you this product with an appreciable yield of polyphenols, and some of them are present at what you might get in red wine. And we're interested in the still beans, particularly I think people generally are interested in still beans and quercetin at the moment at least. And we can see that these are indeed there and stable. So all of this finished right at the time that Debbie and Paul Zalisco and colleagues were successful in getting this FedDev grant. And so as part of that FedDev grant, it will fund this project to go through to completion, which is there's already been equipment purchased, so this industrial scale sonication system that is flow through and refrigerated and has a variety of properties that make it more what you would use for food grade work. And so that's now happening. The process is being slowly commercialized. And then, sorry, that was online. And then the other thing that's really important is that we have, as we should, you know, as a high-profile grape and wine research institute here, should have the ability to do this. There's a ton of companies, more and more every year, that are harvesting their grapes and need to know, you know, not just what it tastes like, but what's in there, and even what's in the pomace, what's in the garbage. You need to know what you can about the material, and so you can make good, informed decisions. And so this is now, with the recent grants, these are now things that will be done here. So we're that FedDev grant is recent, so we've just made this purchase, and we're just starting to work out the methods that we'll use here. And when I say we, I mean Sufen is doing this here. And so Sweet and Sticky will be able to take that basic method and make it work in a way that is appropriate for a commercial application. So all this work was actually done by Shihab and Sam McGowan, who've now graduated and are doing other things. And these are our contacts at Ontario Genomics who have been incredibly helpful in guiding the metabolomics part along the way. And these are the people who were doing the measurements at the facility in Alberta. And that's it. Thank you very much for your attention.
Any questions? Technique that you're using there, is that for the idea of just getting the water solution and precipitating them into a solid, or can you use that to suspend those?
It's in suspension. So beta-cyclodextrin is in solution. You can get, I think, up to 1.8% solution of beta-cyclodextrin before you'll get precipitation. I didn't mention it, but one thing that we played a little bit with was you can also put extra. So you could have a 5% beta-cyclodextrin solution. And as long as you're constantly sonicating that, you've got precipitation forming, it's probably cycling through, and you might load up more. Eventually, eventually it gets freeze-dried, it gets lyophilized, so the whole thing that you get at the end is a powder. But the idea is that you'll load up the cargo, and it will be in solution, the way we did it, and then you'll freeze-dry that to harvest from that solution.
Is that being used in like brewing to get more alpha acids suspended in solution, like anything that's hydrophobic?
So again, you might have noticed it's not really my area. I'm taking something that I did do in this area, and I'm giving you that story. And I know that beta-cyclodextrin has been around for 30 years, 40 years, and it's been used for all kinds of things. And it's that core molecule, beta-cyclodextrin, has been modified to improve hydroxypropyl beta-cyclodextrin, to improve water solubility to you can get a 70% solution. And that one is used to deliver drugs. They are used sometimes to actually mask bad smelling or bad tasting molecules, because often those are also hydrophobic, and so they'll do a good job of bloating those up. Doesn't seem to have a taste. I don't think it has an actual taste. So it's been used in a lot of different food applications, which is how you get to that GRAS designation, generally recognized as safe, because it's been used so much in so many different contexts and there's never been a problem. Diarrhea is if you have a lot of it, but of course that's true of many things, right? So it has no really overt toxicity. So it is definitely used by people making beer, but by people making all kinds of things. I think even when you buy, you can buy polyphenol enriched orange juice, or at least for the time you could, and I think they were using it for that kind of thing as well. And if not, beta-cyclodextrin, things like that get used. I'll get you in a sec, Brad.
So once you bound the polyphenol into the base dextrin, if you ever, and you're using it as, let's say, something you just ingested, that's good.
Yeah.
But if you wanted to re-extract or re-release the polyphenol from the base of dextrin,
how do you do it? Oh, you could do it either way, I guess. I mean, that's the problem they had at the analysis step here. And what they were doing is putting it into organic solvents, because then it's fine, right? Because now you're just doing the analysis. And so you'll extract it in, that's a reversible reaction, and so you'll extract it from that. So you could do either. For analysis purposes, organic solvents, no problem. But certainly there are, I can't remember if it's an alpha-1,4 linkage of the glucose subunits. I feel like it is. But you can use the enzymes that would digest that equally. That's a good question. Brad?
Just wondering, could you improve it? What sort of increase in scale you're anticipating from bench scale to now almost commercial scale?
Yeah, so you can imagine my bench scale thing was the basic kind of instrument that our sonicator that you would have in a regular biochemistry lab. You got your cells or some tissue that you need to disrupt. And this new one is it's a flow-through system, so it's this continued. You've got a chilled reservoir where all the material is, and you're cycling it through. So the volume, I think it takes up to 5 liters of material. The power is much higher and adjustable. So yeah, I mean, from a $2,000 piece of equipment to about 15 to 20,000. It should improve the yield, and it'll all have to be worked out again. Okay.
So thank you very much, Dr. Stewart, for your presentation and for the discussion afterwards. I'd like to invite everyone to join us on February 24th when the lecture series resumes with Ron Jackson. He's going to speak to us about sparkling wine at that date, so we hope to see you then. Thank you.
Yeah, still in order.